Okay, thank you and good morning. Now for something completely different. I have been arriving last night or last afternoon, and I saw really cool talks about the universe and describing the universe in terms of information content. Now there is biology, of course, there is the brain. We saw that in the last talk. And I'm not so much going to talk about brain science. I'm not a brain scientist, I'm a physicist. But I would like to discuss a little bit how to go from a certain degree of understanding of the brain to computers that maybe work a little bit like the brain. Before I do that, I'll show you a computer that you all carry in your bag, in your pocket, and this is a supercomputer carrying out a couple of billion multiplications per second. And of course, what you all suffer from are these three things that I have written here. One is you have to charge your battery continuously, at least once a day. You have to update your software, and maybe it's not so much for you, but for the silicon manufacturers, they have to produce billions of really well-working transistors. Only very, very few can fail. And you guess already that in your brain, the situation is totally reversed. You don't charge your battery, you don't update your software, and in fact, your neurons fail all the time. So the question is, before we go into the brain, what are the computational principles of telephones? And of course, the computational principle is the Turing machine, which is a multi-state system, a system that has a finite number of states, and it can carry out actions on a tape, an infinitely long memory, which collects symbols, okay? And symbols, and that's an important thing here, represent physics in a way. If you want to predict the weather, the symbols will represent pressure, temperature, and other things that represent physical quantities. The action is to change the state, rewrite the symbols, move the hat that reads and writes on the tape, and that's what you call an algorithm. And so there are symbols and algorithms on your cell phone or on any conventional computer. If you look to the brain, definitely energy charging is replaced by eating and drinking. Software is replaced by learning. It's probably the single most important thing that distinguishes us from computers, and which is often forgotten. You and me, and we all, even without drinking alcohol, lose about 1 brain cell per second. That's 100,000 a day. If you would do that to your microprocessor, it would start failing, and not just failing a little bit, but failing fatally after a couple of seconds or so. So that's the resilience or the fault tolerance. Now, it doesn't mean that we learn from biology. That's often the idea is to say, well, biology is great, so let's just copy biology and we will have wonderful technology. Now, that has been criticized by many people. Among them is Frederick Jelinek, who made this famous statement, which is now often made against our work, I have to say. It says, airplanes don't flap their wings, okay? And so engineering has found completely different ways of flying, completely different from nature. And so engineering is the way to go and not looking at nature. Now, of course, that's not quite true. I show you these two pictures here of a seagull and the plane, and you immediately see that they are basically identical. The principle of flight is dynamic lift, okay? That is air flowing along the wings, and there are pressure differences generated by many mechanisms, and both concepts here, the gull, the bird, and the plane fly according to the same principle. Of course, there is something in addition, which you see on the plane. There are the jet engines, which push the plane forward, that the bird achieves by flapping wings, but the principle of flights is exactly the same. And humans would have never invented airplanes without looking at birds before. So again, the question is, what are the principles? Okay, not copy bit by bit or cell by cell, but find out what the principles are. So what are the principles, the computational principles of the brain? The answer is we don't know, but I will give you some ideas. And I'm saying that there are two types of principles. First of all, there are what I call structural principles. That goes back to people like Ramón y Cajal, who has been a neuroanatomist at the turn of the 19th to the 20th century. And he made these wonderful pictures there. These are hand-drawn pictures made from things he saw under a microscope. This is part of the red cortex. And you see cells that are connected by nerve fibers, dendrites and axons. And there's an interesting thing. Cajal, if you look to his drawing, this is his original drawing. He actually puts these little arrows there. And I mean, he already guessed that there is directionality. He couldn't really measure that because these are dead cells. I mean, he didn't really do dynamic measurements, but he guessed that information flows through this system. And so what these structural principles tell us is there is, first of all, local spatial integration. So there is a cell body which receives lots of inputs through tree-like structures, just adding up what comes in. Then there is directionality, there is definitely connectivity, and he also saw that there is some degree of hierarchy. Now that is exactly what people have copied into what's now called artificial neural networks, where often people say it has nothing to do with biology. Actually, that's wrong. It's just a copy of biology. It's a copy of aspects of biology. It's not an artificial brain, but the connectivity, the directionality, the local spatial integration, and the hierarchy are copied from brain circuits. And we know what a brain cell or what an artificial neural network looks like. Effectively, it's just a summing unit. Very simple. And this is summing in space. So this is a spatial distribution. These are the inputs. You may call them dendrites. So there are many here. And then in addition to the summing, there's also multiplication with weights, with real numbers that give importance to the input. And then very importantly, there is an activation function, which is some kind of a nonlinear response. It could be a step function, logistics function, or any other thing. Now what people have done is to build hierarchies and then networks that shift information from the input to the output, and they do amazing things. You know that. They play Go, they recognize images, and they develop into what we call artificial intelligence today. And it's a huge economic impact already. So clearly copying aspects of the brain has been important, but there are some dark points here. First of all, learning in this system requires large labeled data sets. There are companies out there that do nothing else but labeling data sets, looking at images, having humans that look at the images and say, this is a cat, and then label this thing as a cat. All right? So this is what we don't do in our brain. You don't need these kind of labeled images. If I just show you a weird animal, you will kind of recognize that it's weird, but probably even one occurrence is enough. Okay? So these large data sets are not necessary. Also, in our brain, we don't have a procedural separation between learning and inference, which you do in the neural network. In neural networks, you train, and then you use. And we do things continuously, both at the same time. So there is no continuous learning in these systems. And so there are discoveries to be made in the brain architectures. Now there's a second type of principle, which is the temporal principles, and they are not taken into account in current neural networks. Of course, there is recurrency, but there is nothing like continuous time that is flowing where you just follow the laws of physics, how they evolve in time. So time and temporal integration is very important, and very shortly, let me show you how this works. These are two neurons. They look kind of weird. This is a presynaptic, postsynaptic neuron. This is the gap in between. There are neurotransmitters flowing across this gap here. And this thing does integration, not spatial integration, because there's just one input and one output. There's nothing here in space. But what happens is if you have just a presynaptic recording here, which is one of the so-called action potentials, then the receiving neuron shows this kind of response. If you have 3 pulses on the input, bang, 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 all on the same neuron, then the system builds up because of intrinsic time constants. The membrane potential here builds up, and at some point there is this nonlinearity region, which you may call threshold, and then the spike is being generated. And the spikes, that's interesting, are all the same. There's no information on the pulse side. It's all in the timing. So what is timing good for? There are various things. One is sparse information coding. The other is all kind of learning mechanism, short and long-term plasticity, and then there is computing based on temporal noise. And the assets that we get, I hope, and I think we can show that, is energy efficiency, scalability, and also computational advantages. Now, if you want to do work on this, I think what I claim, you cannot speculate. You can always speculate. You can have theories, you can have philosophical insights, but it doesn't help you. You really have to show that things work. I'm an experimental physicist. And my claim is you always require a model system to test your ideas. And what you do with the model system? Well, 2 things. You can help understanding the brain, biological information processing, and you can develop what we call future computing, computing that works a little bit like the brain. And I would claim there are 2 different modeling approaches. One is what I call numerical modeling, Turing machines, where the parameters, as I said, are represented by symbols and the actions by algorithms. So if you have a membrane potential as a voltage, you don't represent that by a voltage, but by a set of symbols, like zeros and ones. Then there is not Turing, non-Turing, the physical model where information is represented by physical quantities. So voltages, for example, represented by voltages, and the actions just by physical laws. And of course, there are combined systems. So what you can do, you can first start Turing. Okay, you can say this is a differential equation that describes the membrane potential as a function of time. The cell has conductances, capacitances, and the interesting thing is if you divide the two, C by G, you get a characteristic time constant for the membrane capacitance. For the more electrotechnically oriented, this is RC. Okay, this is the voltage on the membrane, and then there are these horrible sums. And these sums extend over tens of thousands of synaptic inputs, and this is the kind of network that actually people then simulate Henry Markram on supercomputers. They put them on chips like that. This is a neuron, but there is no neuron here because this is a supercomputer. You represent the neuron by symbols on the Turing machine. Now we have a full dome movie. I really like this technology here of a neural network. Can we see this, please? This is work done by the group of Markram at EPFL Lausanne. And this is one of those networks that they actually reconstructed. This is not real. This is computer reconstruction. This here is a cell body. And you'll see that there is a lot of connectivity. There are many inputs into the cell body, and there is one output, which is the axon. And what you see, there are all these connecting points. Here, for example, and here. These are so-called dendritic spines. And these dendritic spines connect one neuron to the other. In total, you typically have tens of thousands of connections per second, per neuron. Okay, so let me go back to the slide that I want to show. Now, if you run these simulations, what you find is that you can draw interesting conclusions, but there are some drawbacks here. One is that the simulation typically runs 1,000 times slower than biology. And it runs about a factor 10 to the 14 more energy per operation. What's an operation? Well, for example, the simulation of a synaptic transmission. Is that a problem? Well, energy-wise, it means for a full brain simulation, you will need five times all electrical power that is generated in German power stations, for example. This is my reference here for a full brain simulation. This is hard, probably impossible and useless and silly. On the other hand, you can say, well, in principle, it's about understanding the brain. Maybe humankind should build these huge power stations, should build these huge computers, and then we understand the brain. But there is a more fundamental problem. You need 100 years to simulate a day of learning, okay, or 1,000 years. And that is a severe problem because learning is a slow process. It bridges different time scales. And also, you want to repeat experiments. You want to see, you have a model, you test it, and you want to test another one. So we need thousands of years to do reasonable learning experiments. And so this really calls for new ideas, and we need new, what I call brain-like or neuromorphic computing architectures. So what's the idea? The idea is very simple. You have this equation, and rather than putting it on a microprocessor by using symbols that calculate, you just do a simple thing. You do a physical model. For a capacitor, cell capacitance, you buy a capacitor, you buy a battery, you buy a resistor, and you wire everything up. Of course, you may call this analog computing, and analog computing is known to fail because of lack of scalability, but here the communication is happening through these spikes, which are binary. So in principle, this is what we call a mixed signal system. This is a very technical slide. It's an actual implementation of what we do on these chips that we build in the Human Brain Project, which is a neuromorphic chip. And you see circuits here. And you see this kind of circuit that I have shown. It's all the analog integration of information. And then there is this blue triangle, which is a comparator. It poses a threshold and measures the voltage on the capacitor. And if it passes a certain threshold, there is a rectangular spike being generated. That's being sent into the network. It's then shaped so that you really have this kind of postsynaptic potential that can be integrated in time and in space and then sent back to a receiving neuron. This is one of our chips. Now, what I would like to show you a little bit is how these chips look like. Okay, this, you know what this is, probably. I don't know. This is a transistor. And a transistor, as you remember from school, it has 3 legs, source and drain, and a gate that influences the current underneath. And what I want to show you is, because I like technology, as you may have noticed, another full dome movie. Maybe I had a big effort with the people here, and I'm very grateful for their support. What you see here is, so start running it, is it doesn't really fit because all those should be rectangles, and they aren't rectangles. But what you see is sort of a micrometer type piece of the chip that we built. And you may recognize there are transistors here. All these are transistors. And what we do in our research, we wire them up to build brain-like structures. And we zoom out now. Now we see many, many micrometers. And what you see all these transistors here. And if you zoom out, you start to see regular structures. Wait a little bit. Now you start to see there are repeating structures. Okay, this is maybe like 50 by 50 micrometer. And those repeating structures are synapses. These are all synapses. So on a chip, there are about 100,000 synapses. There are more and more coming up now. This is like a millimeter or so. Now you see some large structures appearing, which make the connectivity between the chips. They are huge, like many, many micrometers. But you see all the synapses. See the synapses. There are hundreds of thousands of them. They really make up the information processing in the brain. Now see the borders of the chip and the back of your, but it should also appear here at some point. You see the chip borders. They are bent because of the dome. But these are the locations of the neurons here. So on a single chip, we only have like 500 neurons, but 100,000 synapses. So the neuron, or the synapse to neuron ratio, is really large. And this is what we have in our brain. The synapse to neuron ratio is 10,000, and this is exactly what we have on our chip. Now you see many, many chips, like hundreds of chips. The flickering is just an artifact. I'm sorry. And what you see now is a wafer, a complete wafer. Now the thing has moved back to the first slide. I don't know why this is. And what you have just seen, of course, was not a real wafer. It was a computer animation of the chip design that we submit to the company. And this is the real wafer now. But that's what you just saw. Those were the synapses. These are the neurons. And this is a complete wafer, 20 centimeter, which carries like 200,000 neurons and 50 million synapses. There is an interesting aspect here. Why is this better for brain simulation? Now, these are analog circuits for integration. And I said there is RC, or C divided by G, the conductance. And that's the timescales. Now, of course, there are many other timescales. But on this system, all the timescales are scaled down by a factor 10,000. So a day, rather than 1,000 days of simulation, really only requires 10 seconds. So it's a time-compressed system. This is what it looks like. This is a single chip, not a wafer. But at the end, we use wafers. This is one of the units that we built in the Human Brain Project. I just told you the numbers. And this is the machine that's currently operating in Heidelberg, which contains 20 of those wafer modules. There's a control computer. And you have millions of synapses and millions of neurons and a billion synapses. So what can we do with this? First of all, is it worthwhile? Some references. The energy needed now for synaptic transmission, which on a supercomputer, Henry Markram simulation, is a joule, 1 joule, and which in your brain is 10 to the minus 14 joule, or 10 femtojoule. On the neuromorphic system, is like 10 to the minus 10 joule. So it's a huge gap between both, between biology and between conventional computing. But of course, it's already a big step towards more energy efficient computing. A factor 10 to the 10 is certainly very, very useful. More important is the time needed for a day. Time needed to simulate a day. On a supercomputer, that's 1,000 days. I said that was a slowdown factor of 1,000. In your brain, of course, a day is a day. That's the definition of a day. It's real time. On the neuromorphic system, it's 10 seconds. And that, I think, is really the main step for progress in the field. Now, what can we do with it? I give you 2 examples. There are many, many. If you want to read them, I give you a reference. But these are just 2 application examples. And the first one is what I would call a reverse engineered biological circuit. And it's a deterministic circuit, I should also say. It's about insect olfaction. There are insects. They can smell flowers and identify them. And they do that by a technology which in machine learning we call multivariate data classifications. You have many inputs, which are chemical sensors in this case. And then they are combined, and they say, well, this combination corresponds to a tulip, because we are in the Netherlands, or a rose, or whatever. Okay, so you can combine chemical inputs to identify flowers. And I show you this example because it shows you a little bit about the way we code information in this system, because this is about information science. This is time. In fact, this is biological time. You should divide this by 10,000 to get the wall clock time in the machine. This is neurons, neuron numbers. It's a small network, like 200 something neurons. This is the input. This is the output. This is in between. And the dots are firing. Okay, you take this neuron, it makes tuck, 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 tuck. It's like a radioactive source. Okay, and in here, this is the input. And you see the input looks random. There seems to be no information. And in fact, if you plot the rates, just the rates, it's a flat histogram. It doesn't show any information. Look to the output, also no information. If you do the training now, the classical training of a neural network, the input is still the same, but the output suddenly shows peaks occurring here, which means the insect or the chip has recognized something from the input. And it's quite interesting to see that the information here is coded in rates, but it's not clearly visible. And also, you'd immediately see that this is very expensive in terms of energy, because you have to pay for each click. Okay, this is called rate coding. This is what insects do. This is also what we do in our brain. Also, the output is rate coding. The recognition takes place after 200 biological milliseconds. It's still kind of random here. And then suddenly it has taken the decision that these two neurons should fire to recognize a certain flower. The cool thing is, look at all these neurons in between, which is the majority of them. And in large networks, this is even more extreme. They almost never fire. In particular, after learning, it even gets less. But this is where information is processed. This is where we do the recognition, where the learning actually has an effect. And you can do this recognition with very sparse firing. And this is extremely energy efficient. Information is coded by the timing and not by the rate, like here or there. This is expensive and stupid, but it's the only way to interact with the analog world or with the classical world. Here you have time-based coding, which is very efficient. The second example I would like to show you is more like a physics information theoretical approach. It's the Boltzmann machine, where we now suddenly make neurons stochastic. We have these networks, and we build a Boltzmann machine by having symmetrically connected stochastic nodes. And the state of each node, this is a neuron, is described by a vector of binary random variables, which are 0 and 1. How can that be? Well, for example, we can say a neuron is active in sampling, or it is refractory, which happens after a spike. These are the two states. So it's a physical two-state system. And the probability of a state vector, that's easy to show. It converges to a Boltzmann distribution. Okay? And the Boltzmann distribution, of course, has this exponential term. You know that, and it has a state vector, or a state function. And this is, of course, represents the energy function that's being learned. This is like in a magnet. Okay? So you have this interaction between the nodes. These are the weights. And this is some kind of a bias term. So what for? To learn internal stochastic models of input space, generative or discriminative. Let me first shortly comment on how you produce stochasticity. How do you make a neuron stochastic? Is it intrinsically stochastic? Some people say, well, biology is kind of warm and soft. There are thermal effects going on. It's all fuzzy and not sharp. That's wrong. The biological neurons are totally deterministic. They are super precise elements. Still, if you record from a monkey that does something useful, and you record a neuron, it's totally stochastic. It looks like a randomly firing thing. Why is that? If it's singular, if you take out the neuron from the monkey, put it in a petri dish, it's deterministic. So clearly, it's the background. And we made this kind of model, which we call the sea of noise, okay, which emulates in vivo brain states. So we have a big network, and they have these subnetworks here, which are all Boltzmann machines. They all do something interesting, like recognizing faces. And then we feed what this system does to this one. And this one interprets this as noise, because they are doing different things. So if we have this sea of Boltzmann machines, we produce stochasticity by actually functional activity. That's a pretty cool idea, and it seems to be realized in biology. Now, how can we show that really learning produces probability distributions? If you take decisions, you sample from a learned probability distribution. There's this famous thing here, which you may have seen. This is this strange animal. And you sample now, in this moment, if you look at it, from the probability distribution that you learned about animals, and you flip, this is bimodal distribution, you're not so sure. In most cases, it's clear. You say this is a monkey or an elephant, no problem. But here, your probability is tricked a little bit. You switch between a monkey and a duck if you train. And there's some kind of switching time, and we try to do the same thing. So we train the network by some clever mechanism, which is called the wake-sleep mechanism, and then we can do two things. And did this network learn to recognize numbers, by the way? Now, what you can do is you can close your eyes, and you still see things. And you infer from learned probability distribution without actually having input. You may call this dreaming. And we can do this with our system. So we train it to see numbers, and then we switch off the input. We let the system free running, dreaming, or more scientifically, generative mode. There's a paper here in the physical review about that. And these are just 3 numbers which the system had learned, very simple. And what you see is a 0, a 4, and a 3. And it does what you do up here. You switch between these kind of solutions. And our system also switches between those solutions. This is what it dreams. And sometimes it's in between. This is the path it takes. In most cases, it sits at the 4, the 3, or the 0. But the numbers it shows are not those that it has learned precisely. Those are numbers generated from features that it has learned. So those are 9s and things that it hasn't seen before. But it looks like it thinks it's a 0 or a 4 or a 3. Then you can do inference. That is what you really do when you live. When you open your eyes, for example, you get a constraint that the middle bar is always on. And then this is incompatible with the 0. And then you see the switching is only between the 4 and the 3. And that's quite interesting because you can also measure things like switching time. You can measure the switching time here, your switching time. You can measure our switching time. Interestingly, they both follow a gamma distribution. So there's a lot of really cool theoretical physics behind this. Before I close, neurons are more complicated than what I just showed. They're not just integrators in space and time, points that integrate in space and time. They actually have a structure. And in the Human Brain Project, we have really cool people, like Javier de Felipe from Madrid. He works in the former lab of Ramon y Cajal. You can see him as a successor of Ramon y Cajal. He is now using electron microscopy. And they analyze neurons. They really look at them. This is actually a human neuron. It's one of the ones we have in our brain. And they looked at the morphological features. Like, these are the dendrites that integrate information. How long are they? What is their connectivity? How many synaptic spines are there? So they really measure these things and provide a whole lot of data. And what we have done together with Javier and also theorists like Walter Senn in Bern in Switzerland, Matthew Larcom is an experimental neuroscientist doing in vivo measurement in Berlin, we developed a theory which I call biological learning. It's a cortical circuit for error coding. So we go away from this idea that we use classical learning mechanisms, backpropagation you may have heard of, but we just do the continuous learning like in our own brain. So what's the idea? The theory is that these extended neurons, these dendritic trees, really have a very cool role. So the upper part here receives feedback from upper brain areas, from higher brain areas, what you have already learned. So this is a prediction. So the neuron here, this is the cell body, continuously predicts what it expects to see. If I look out there, I expect to see people in certain faces. And there's a constant influx of prediction. Then this is reality. This is the external feed forward. We call this the proximal input. Okay? So there is constantly an input from what you expect and what you really see. And then there is this middle part here, which is the apical trunk. And it uses some calcium-based mechanism that really looks for a coincidence between what you expect and what you see. You know, if I look there, I see my computer. This is what I expect. If I would see an elephant there, I would be surprised. It's not what I expect. And then this neuron would say, well, there is an error. You should have seen a computer, but you saw an elephant. And there's a prediction error which we feed back. The soma, the cell body, codes this by activity. And then this part rewires. So the next time I look there, I know that there's an elephant. Okay? So this is continuous update of our knowledge. If we only see things that we expect, nothing happens. We don't learn. We don't learn if there's nothing new. But if there are new things, and there are not too many new things in our life, then we constantly update our knowledge. And if you're a little bit into neural networks, there is this backpropagation mechanism, which is basically calculating gradients towards a good solution. Many people said this is not a biological reality, it's just a mathematical mechanism, very, very successful, because it's sort of the backbone of deep learning, but it has nothing to do with biology. Now, this mechanism suggests that backpropagation is actually a biological reality, that it is implemented in biological circuits. Now, this is measurement, real measurement in biology. There's real theory behind this. Look for a paper of Walter Sen. It's really very heavy mathematical theory, which I don't do here. But we do something else now. Of course, you can put this very heavy theory and all these very complex dendrites into mathematical models again. And then your factor 1,000 of slowdown gets even worse. We tried it. It's more like 5,000. To simulate dendritic integration is very, very hard. So in the Human Brain Project, we made an effort to implement dendritic integration in what we call the second generation. 2018 BrainScales is the name of our system, full-size prototype chip. And this is it. It looks like the old one, nothing special. But it's interesting now, some numbers. It has 60 million transistors, which may not impress you. There are chips with billions of transistors like graphic cards. But the interesting thing is out of those 60, 30 are analog 30 million, full custom design, really hand-drawn, like the thing I just shown you. They sit here. And now they contain all the dendritic integration. They have so-called compartments. We don't talk about neurons anymore, but about neuron compartments. And we can have any dendritic structure. It has all kind of learning mechanism. There's a little cheat we do that these are classical processors, which we now also put in. Each of these chips has 2 RISC processors, which allow us to run software-based learning algorithm, those things which we cannot yet put in hardware. So this is a cool chip, which is now somewhere the design is in Taiwan. It's being produced by the company TSMC, that also makes your telephone chips, by the way. But now they produce this thing for us, and by the end of this year, we should have it operational and then really show this kind of continuous learning. I'll end by showing you this thing, which is another pretty picture. These are neurons. These are the neurons are these long structures, like 300 by 20 micrometers. There are about 300 transistors in a neuron. And I show you this because, you see, by working with biology, you can do cool stuff. I said we had to switch from point neurons, as we call them, to compartmental neurons. And we discussed with them what to do. And in principle, all you have to do is to put little programmable resistors between those neuron circuits. And then we found out that this is basically the old chip. All we had to do is to add this little row here of transistors. Those are the programmable resistors that can connect these things, which we now call compartments, to really build dendritic branching. So this is how, it's a very nice demonstration of how really sort of the physics of biology, the physics of neurons, can be directly mapped to analog neural circuits on silicon. Now, if you want to know more, see the real thing, even try for yourself. You can register. Our systems are accessible on the internet. You can register as a user. At the Human Brain Project, there is a very simple website, neuromorphic.eu, where you also see what we call the guidebook, which shows you all the technical details. I could not discuss a lot of technology here, but there is a lot of technology, believe me. And this is all nicely described here. And there are also some exercises you can do on your own computer if you don't want to use hardware. If you want to get into hardware, let us know. Thank you very much.
Thank you very much, Karlheinz. We have time for a few questions.
Yes, you hear.
So where do you see this going in, say, 10 years' time or 15 years' time? Are these going to replace, what's the application in a more general sense? Is there a particular area where we'll just replace, your mobile phone will be full of these things and they will do stuff that we...
Yeah, I mean, that's the number one question we get asked, and it's a very reasonable and necessary question. The application is in an area that I call cognitive computing. It will not replace Excel. It will not replace your email. It will not replace Excel to calculate your payslip, obviously. You don't want this to be stochastic. So there are certain things computers will always do. Classical computer, there's no doubt about it, they will get better. But cognitive computing basically means that you continuously learn. You learn new input, and you use this to make predictions. I see those things as prediction-making machines where you detect surprises. You say, there's something interesting going on. What should I do with it? Should I learn it? Or should I just alert you that something is going on? So I expect that we will see these kind of neuromorphic systems as part of one of these buzzword things, the Internet of Things. where we have many, many sensors that measure all kinds of stuff and currently produce a lot of data, totally useless data. That's being sent over networks. It congests our networks. It costs a lot of energy, but nobody looks at this data. If you have a sensor that measures, I don't know, whether something is wrong in your car, vibrations or something like that, you don't want this information if everything's okay. So you want local processing, you want local intelligence, and the car should only say, here, there's something wrong with me. Please look after it, but it will only happen once every 10 years or so. So it's a massive data reduction by local intelligence. That's where I see the #1 application.
Fantastic talks. I don't really have a question, just a request. Would it be possible to play again the full dome visualization of the neural network? Because it's not under my control. But yes. Please play. Whatever. That is the network. That is the Henry's work. I should say Henry Markram. We initiated the Human Brain Project together. Can you switch off the computer? This is darkness. Yeah. So this is the typical. There is no activity shown here. It's really just this structure. There are also movies with activity which you can see on their websites, and it really gives the impression on the density. This is a realistic density that you have in your cortex. Actually, it's a 6-layer cortical structure of a rat, which is reconstructed here. And there are the big blobs, which are the cell bodies, where the information processing that I described, the conductances and the capacitance are mostly taking place. But of course, the cell really extends along those things here. Also, those have a membrane, and activity also spreads here. So it's not like in the classical neural network, where the information from here to the cell body travels basically in no time. But there is electrodynamics that actually describes the activity along those dendrites and axons. So there is a lot of biophysics there. And the question is, do you really want to simulate all the biophysics? And Henry and his group, their approach is, yes, we want to do this, because he says once you have to do it, and then you can reduce the complexity and see what are the key features. And this is the key features. Those are the ones we now get then for our chips, because we cannot put in all the biophysical detail. But we say, well, maybe there are these steps, these are compartments, they have certain features, and those features we put into our hardware. So there is complexity reduction from this super precise model to the chips, which is substantial. And the question is, how much can we forget, and how much do we have to implement? Where is the border? And that's a research question. That's not finally answered. So these little blobs there are the synaptic connections. The synaptic connections are along the dendrites. And as I said, for a typical pyramidal neuron, like these layer 5 neurons, what you saw in real life there from Javier's De Felipe's lab, that typically has 10 to 30,000 synaptic inputs. And so this matching of prediction and feed-forward input is really based on very complex structures. And if a simple neuron, not a simple, but a single neuron can in principle code an entire image, which is really means a neuron is a computer. It's not just a simple integrator. Okay, I think we can stop this. Otherwise, I get drunk here. It's very hard to stand actually if you look at these things.
Time for one last question.
Well, you partly approached this in the answer you just gave, but the work you're doing is great. But I'm wondering, have people well, actually you've considered it, but if you have any ideas on why, whether you need the architecture of the brain to reproduce intelligence or animal behavior in general. In other words, is it possible that there's a totally different topology that would yield that would yield intelligence? Of course, we know that the brain, as you say, is 10 to the umpteenth times more intelligent than more efficient in processing. But are there other approaches to obtain what we know as intelligence or behavior rather than?
Yes, there's always reality, and there is what I would call speculation. The good news is that we know a system that works, and that is the brain. It has all these amazing features, and we now, by really huge effort experimentally and theoretically, start to understand aspects. So it's the obvious thing to use this as a case to implement. There may be other ideas. The question is always, is it worth to invest into these things? Because ideas need to be somehow grounded. I'm a natural scientist, a physicist. They have to be grounded in real knowledge, I think. Yeah, but if somebody comes with a totally strange and weird idea, one can certainly look into it. The question is, would you make the effort to implement it? Because doing things like this costs you 10 years of your life. It's a decade. And before you start investing, you want to be sure that it can work in principle. But I'm always open to new ideas. But I had a rather limited view. I say, this is nature. Nature works. So let's try to understand, which is number one thing. And second, let's make use of it.